This will be just a very quick overview of the Vandenberg paper. One of the advantages of a class like this is that you don't have to buy a textbook. The disadvantage is many of the papers are fairly technical. This one's no exception. There's a large part of it that's based on the mathematics, the statistics, and I don't expect you to learn that. I don't expect you even to read it all the way through. So typically the best way to approach these papers is to read the abstract in some detail. Maybe if there's an overview section, skim over it. And then these guide videos should help to guide you and take you through the major points. Okay, so much of the important things we already talked about in our previous lectures, but I'd also like to look at a couple other things they bring up that are useful. So on this first, I guess, second page, there's an overview of the process, which we've already talked about. The only thing I want to add to this is they have a stage here where they are looking at enriching the RNA targets. Now, when they say enrich, basically what they mean is they are using an enzyme, a molecular biology process, to amplify the total number of copies that are there. Now, realize that if we're going to do a differential expression study, that we need to amplify them in a non-biased way. If it's biased in any way, then that's going to throw off the initial amount of our transcripts that were present in the sample. So many stages or many protocols do require that we enrich the samples because there's just not enough to start with to get an accurate measurement. And so if we can amplify them in a non-biased way, that's fine. Everything works great. And the rest of this we're fairly familiar with. The data analysis part is rather large, and we're only going to give you a little bit more of than what we've already talked about. All right. The next thing I wanted to go over, there's some stuff about sequencing and read length and how that affects. That's not really critical. But the next major thing I wanted to hit on this is looking at the difference between a transcript. I've been using the term transcript just because it's a little bit more general, but they also they point out that some studies do gene level estimates of abundance rather than transcript level estimates of abundance. And you're like, wait a second, I thought transcripts and genes were the same thing. Well, not necessarily. Number one, a transcript could refer to something that is transcribed but maybe never fully translated. So depending on your definition of gene, we could have transcripts that were present in the RNA form but then never were translated into proteins. But more importantly is the realization that in many eukaryotes, there are many genes where a single transcript can be spliced together in multiple ways. And so although we might have one gene, there might be 3 transcripts that result from the transcription of that gene because of differential splicing, alternative splicing, sometimes it's also called. So here's an example where we have a single gene, but it can be spliced together in 3 different ways. So notice the intron number 1 is the same, and intron number 2 is shared between the blue and the red, exons, I should say. Exons number 1 are the same for all of them, but exon number 2 is the same for blue and red, but different for green. And then exon number 3 is the same for blue and green, but different for red. And so we splice those together and we get 3 slightly different versions of that gene product, but they all come from the same gene. So if we were doing gene abundance estimate, we would count all the reads that map to all of these the same. And if we were doing transcript level, then we'd do 3 different ones. Now realize this can really complicate our ability to figure out exactly how many they are, because reads, for instance, if all of our reads are coming only from this transcript, we don't know whether they were there for transcript 1, 2, or 3, because the exon number 1 is the same for all 3 of these. And so we have a much more limited database on these transcripts that are truly unique. So notice the ones that they've coded with blue, red, and green can be assigned to all 3 of the above. And then if those transcripts are there at very different amounts, we don't know that maybe 30% of these are from one transcript and 70 or 60% are from another, and only 10% are from the third. And so we would need to find ones that can be uniquely assigned to one transcript or the others. And there are some statistical inferences along with models that we can do to maybe get a little bit of information out of the ones that are potentially all 3. But it becomes a much more complex process when we're trying to do transcripts and not just genes. And then the other thing that I wanted to mention is if we have a complete genome, we don't even really have to do the first step of assembling, the global assembly, because we already know what the potential targets are. So for humans, mice, fruit flies, any other model organism, we don't need to do global assembly. We can just start from our original treatments, and then we already know what genes we're looking for, and we can assign each read to a gene. Still, if we're going to do transcripts, we need to understand all of the alternative splicing possibilities, and there would still be some levels of uncertainty if we're doing transcript counts rather than gene counts. So just be aware of that. It's not a precise, we are going to get everything, and there's going to be some level of uncertainty. And so we use statistics to help us understand and evaluate those levels of uncertainty. And then the only other thing there, so we've got a long section here that's kind of about the statistical and mathematical framework. And then there's a little bit about some of the programs that are used also. That's very technical. We're not going to do it for this class. I do want to just say something about the very end here. When we are looking at single cell transcriptome sequencing. This is very, very recent and does require that enrichment stage where we increase the total number of transcripts. But again, if we can enrich in an unbiased manner, it's perfectly fine. So there are a couple different methods, but the basic idea is that we can completely isolate a single cell and then take it through the same process. So we could even look at different cells in the same tissue, or we could look at cells where we have just a tiny, tiny fraction of a sample. And so it really gives us flexibility and a little bit more magnification, right? We can really zoom in to get all the way down to transcription going on at a single cell level.